Bacterial flagella icon of the intelligent design movement. Dr. Alan L. Gillen wrote this in 2015. He wrote of magnificent and miraculous microbes. There are many extraordinary examples of design in the microbial world. In this chapter of his book, two examples are given: the bacterial flagella and the production of blood red pigment in Serratia marcescens. We're getting down to the molecular machinery of the bacteria flagella is magnificent. The amazing ability for Serratia marcescens, a rod-shaped bacterium, to produce a pigment that resembles blood is quote-unquote miraculous. Could not have evolved. Well, we begin with Michael Behe, who made the bacterial flagellum a popular argument for the intelligent design in Darwin's Black Box, his book, using them to illustrate the concept of irreducible complexity. The flagellum is a corkscrew-shaped, hair-like appendage attached to the cell surface, acting like a propeller and allowing the bacterium to swim. Tiny little microscopic things that are alive. The bacterial flagellum is an irreducibly complex process. What that means is it can't have evolved. Somebody had to design it. An irreducibly complex system is one that requires several interlacing parts to be present at the same time, where the removal of one or more parts causes the whole system to malfunction. Destroy one part and the whole system falls apart. Now this tiny little microbe is so much simpler compared to the human body. So tell me about the human body. It evolved? Well, the purported mechanism of evolution, on the other hand, is that a new trait will confer a selective survival advantage and thus enable its possessors to compete better than organisms without the trait. In neo-Darwinism, evolution, a new trait would have to be completely developed. No halfway measures would do. Given this requirement, new features are so complex that neo-Darwinism, gradualism, is very improbable, because an incompletely developed trait would offer no selective advantage. The mousetrap example. Now, Dr. Behe, biochemistry professor, you just widen this up a little bit, and author of the 1996 blockbuster book Darwin's Black Box, has challenged the classical neo-Darwinian explanation that intricate cell structures arose by chance. In this book, he uses the flagellum to introduce the concept of irreducible complexity. If a structure is so complex that all its parts must initially be present in a suitably functioning manner, it is said to be irreducibly complex. All the parts of a bacterial flagellum must be present from the start in order to function at all. According to Darwinian theory, any component that doesn't offer an advantage to an organism, i.e. doesn't function, will be lost or discarded. How such a structure could have evolved in a gradual, step-by-step process as required by classical Darwinian evolution is an insurmountable obstacle to evolutionists. How a flagellum is used, however, adds an additional level of complexity to the picture. You don't just have the feature, but you have to have the programming, the process by which it functions. Some bacteria have a single flagellum located at the end of a rod-shaped cell. To move in an opposite direction, a bacterium simply changes the direction of the flagellum rotates. Other bacteria have a flagellum at both ends of the cell, using one for going in one direction and the other for going in the opposite direction. A third group of bacteria has many flagella surrounding the cell. They wrap themselves together in a helical bundle at one end of the cell and rotate in unison to move the cell in one direction. Come on, that happened by accident, by evolution? To change direction, the flagella unwrap, move to the opposite end of the cell, reform the bundle, and again rotate in a coordinated fashion. It's like they're thinking. How do you figure that one out? The structural complexity and finely tuned coordination of flagella attests to the work of a master engineer who designed and created flagella to function in a wonderfully intricate manner. You might call it the maker's molecular outboard motor. Its most interesting aspect is that it is attached to and rotated by a tiny electrical motor made of different kinds of protein. Like an electrical motor, the flagellum contains a rod, a drive shaft, a hook, a universal joint, L' and P' rings, bushings and bearings, S' and M' rings, rotor, and a C' ring and a stud, stator. The flagellar filament, propeller, is attached to the flagellar motor via the hook. To function completely, the flagellum requires over 40 different proteins. The electrical power driving the motor is supplied by the voltage difference developed across the cell membrane. This motor is one of nature's best molecular machines. Some scientists have called the bacterial flagella the most efficient machine in the universe, with its self-assembly and repair, water-cooled rotary engine, protein motive force drive system, forward and reverse gear, operating speeds of 6,000 to 17,000 RPM, direction reversing capability, and hardwired signal transduction system with a short-term memory. Tell me that's going to happen by accident. Bacterial flagellum paradigm for design in Yersinia, example number 1. After Michael Behe made the bacterial flagellum a popular argument for intelligent design in his book Darwin's Black Box, Scott Minnick joined the ranks of the intelligent design movement. Dr. Minnick is a geneticist, an associate professor of microbiology, University of Idaho. He takes the argument to the next level by describing how the design paradigm led to new insights in his research. Minnick has been studying bacteria flagella for over 15 years and has published work in the following areas: the structure and function of flagella in Yersinia and Salmonella species, assembly blueprints and genetic instructions, detailed descriptions of the transcriptional and translational regulator genes, and integrating motility with single transduction chemotaxis. These are the microscopic pictures of this Yersinia enterocolitica. Everybody's got to have these. I make it simple so we lay people can understand, but I do understand. In extensive research, Scott Minnick has discovered that bacterial flagella provide a paradigm for design. Minnick has been working with the genetics and flagella structure of Yersinia enterocolitica, cousin of Yersinia pestis, a pathogen of bubonic plague, for more than a decade. So this Y. enterocolitica is a cause of foodborne infection like E. coli or Campylobacter. It is commonly found in the intestines of livestock. It causes food infections due to contaminated meat and dairy products. It causes enteric fever and may produce severe life-threatening infections. After describing over 30 individual proteins that make up its rotary motor mechanism, close to 50 in the entire flagellum, Minnick noticed that the basal body of the flagellum produced a toxic secretion when the bacterium was under stress. If Yersinia was kept happy at 20 degrees centigrade, 68 degrees Fahrenheit, and in good environmental conditions, low osmotic saline, the basal body produced a hook and filament, the remaining portions of the flagellum. Minnick had predicted from his genetic studies that a good design would be used for diverse purposes, like engineered design structures that serve dual functions. It is good genetic efficiency or optimal genetic design, minimum cost-benefit ratio. Even before the observing this in humans, he predicted what would happen. Yersinia was quite motile in its environment and could propel its rotary motor up to 100 RPM. On the other hand, if Yersinia were incubated at 37 degrees or 98.6 degrees Fahrenheit, or another stressful environment like high salt, the basal body acted like a cannon, and produced a harsh toxin. Its technical name is type 3 secretion system. It is described in more detail in Chapter 9, the origin of infectious disease. In observing cells from gastrointestinal tract, it was observed to avoid engulfment by macrophages. In its own defense, Yersinia produced a missile to avoid being eaten by human body defenses. The utility of a design model instead of a Darwinian one not only produced good science, but also had practical implications for medical microbiology and clinical medicine. Here we see evidence that design models accurately predict biological outcomes. Thinking God's thoughts after him, and openness to the idea that a creator has made biological structures with purpose is the key to success in biological study. Evidence, not evolution. Got that? Evidence, not wild guessing. Creation, not chance. Design theory works. The bacterial flagellum is truly one of Providence's prokaryotic wonders. Yersinia enterocolytica, biological rotary motor. The sensory and motor mechanism of the E. coli bacterium consists of a number of receptors which initially detect the concentrations of a variety of chemicals. This thing is a thinker in its own way, right? It's programmed. Who programmed it? A piece of sludge without life? Secondary components. They extract information from these sensors that in turn is used as input to gradient sensing mechanism. The output of this mechanism is used to drive a set of constant torque, proton-powered, reversible rotary motors. Now we're talking about a tiny microscopic thing here. No scientist invented this. It's beyond a scientist to even conceive of something like this and then create it. So the output of this mechanism is used to drive a set of constant torque, proton-powered reversible rotary motors which transfer their energy through a microscopic drive train and propel helical flagella from 30,000 to 100,000 RPM. You can't even see this thing. This highly integrated system allows the bacterium to migrate at the rate of approximately 10 body lengths per second. Wow. How fast do bacteria move with their flagella? Some have been clocked at up to 100 per second. Micro something per second. Micro inches, micrometers. Or the equivalent of 50 body lengths per second. Wow. By comparison, bacteria move twice as fast as the cheetah, the fastest known animal. Cheetahs, which run up to 70 miles an hour, go a mere 25 body lengths per second. Generally, bacteria with polar flagella move faster than those with paratrichous many flagella. The complexity of the bacterial flagellum is direct evidence against neo-Darwinian evolution. All the interwoven parts of the body point to an intelligent creator. In the early 1990s, Dr. Michael Behe argued for the intelligent design of the human body. His argument is called the principle of irreducible complexity. To illustrate the complex nature of this principle, one needs to look at the design in driving.